All right, so I was asked to speak a little bit about rare epilepsies today, and as was described in my background, so I'm a pediatric neurologist and epileptologist, and I focus particularly on genetic causes of epilepsy and other similar rare diseases in the neurogenetic space. My talk, not surprisingly, has a little bit of a lean in that direction. But I recognize that there's a broad audience for the Epilepsy Foundation of Colorado and Wyoming. And I'm happy to sort of go in other directions and discuss some other things as well. As we talk about rare epilepsies, because I just want to be clear that in many ways, every epilepsy is a rare epilepsy. Every individual who has epilepsy is different and unique. And there's no two presentations of epilepsy that are exactly the same. So having said that, I had to sort of structure the talk around something. So I'm going to talk a little bit about what epilepsy syndromes are and discuss how that relates to genetic causes of epilepsy and how that informs the way that a physician might approach an individual with epilepsy and think about their case. So I do not have any major conflicts of interest here. I have consulted for a few companies as part of the work that I do, and that is included here. So again, I'm not talking about one particular type of epilepsy. So many of the things that I discuss here may or may not be applicable to you or to your child with epilepsy. And so I just want everybody to take this with a grain of salt, that I'm speaking in generalities to a certain extent, but I'll provide a few examples as well. And then just what I mentioned before, that there are hundreds, maybe even thousands of rare types of epilepsy and causes of epilepsy. And I certainly can't speak to all of them in the time we have today. So I'm presenting more of a framework to how to think about them. But if there are specific questions that come up, I'm happy to try to address them. So this is sort of what we're going to go through. What is an epilepsy syndrome? How do we define epilepsy syndromes? Genetic versus the features or phenotype of the symptoms that an individual presents with. And what do we think about when we're kind of approaching a patient? How do we organize or categorize different epilepsy syndromes in our mind as clinicians? Again, what is the role of genetics within epilepsy, and how has that changed over the last few years, and where is it headed? Including what's the role for treatments and changes in management based on genetic diagnoses as we go forward. I want to point to a couple of good resources here in addition to the Epilepsy Foundation of Colorado, which I'm assuming everybody knows about. There's also the International League Against Epilepsy. Epilepsydiagnosis.org actually has a breakdown of all the different types of epilepsy syndromes that are reasonably well described and can help provide features about those syndromes and how they're different than other ones. So just to sort of start with the basics, what is an epilepsy syndrome? Any syndrome in medicine is a collection of clinical traits or features that are occurring in a particular individual. And when we think about epilepsy specifically, that could be obviously what type of seizure an individual presents with, but also when did seizures start, what age for that individual, were there any associated other conditions or challenges like developmental challenge, what did their EEG look like, and the pattern of the EEG, not just during seizures, but also in between seizures. And then anything else that we call comorbidities, which is really any other signs that there's something else that's a challenge for that individual in addition to their seizures that may be linked overall to neurologic function or even in some cases, non-neurologic function. How distinct is one epilepsy syndrome compared to another? I think it's important to sort of realize, and many patients have experienced a situation where we're maybe not quite as specific as we wish we could be about what epilepsy syndrome a particular individual has. And this is because there are a lot of overlapping features. And being specific about it is the goal, but in some cases it may not be totally clear yet. And so this idea that one syndrome is really unique and separate and different than another one is a little bit better in theory than it sometimes is in practice. And we could think about that by this example. So if the star represents a set of symptoms that a particular individual is presenting with, which type of epilepsy do they have? Do they have a type A, type B, or type C epilepsy? And you can see that they actually overlap all three circles. They have features that might be consistent with any of them, but they fit best into C. And so that's part of where there's a little bit of ambiguity that can come up around epilepsy syndromes. So why do we care about epilepsy syndromes? What's the value of an epilepsy syndrome? And I think the main thing here is that this is how we have been categorizing epilepsy features for several decades now as a medical community. And so there is guidance that can provide us about what is the epilepsy or the developmental prognosis for that individual. Are they likely to outgrow their seizures? Are they unlikely to? Do we expect more developmental challenges if they're a child? Less developmental challenges? What treatments might be best for that particular type of epilepsy? What treatments could be harmful in a particular type of epilepsy? What are the potential causes or etiologies for that individual that might be worth looking into to try to better define what's going on for that person? And just as sort of a simple example that I think even works for the non-epileptologist, these are sort of two EEGs. I think many people are familiar with sort of what the basic idea of an EEG is. And in this case, each of these lines represents a different region of the brain. And this is two babies' EEGs. And on the left, there's an EEG that looks relatively normal and healthy for a baby. And on the right is one that is very abnormal, something we call burst suppression. Both of these babies could have seizures, but one of these, the burst suppression pattern, actually tells us a lot about how likely that baby is to stop having seizures and to have developmental concerns as they get older. And so that really can guide our level of seriousness that we're going to deal with that, how we talk to that family, what information we can give them, what testing we need to do. In one case versus the other one, and in the case of the normal background, we may be able to be much more reassuring to that family and let them know that while they're having seizures, we think that we're going to be able to get things under control and that sort of thing. It's not perfect. There's always exceptions, but this is the type of information that we can get pretty quickly if we can start to understand what type of epilepsy syndrome does a person have. So then if we think about how do we organize those syndromes or group them, there's a number of different ways that have been done over time. And I'll be honest, this is something that we continue to update as an epilepsy community. So this is not just any one way, there's multiple ways. Sometimes we think about it as what's the age of onset for the epilepsy, sort of neonatal versus child versus teenage or adult. Do we think about it in terms of being generalized or focal or both? Or do we think about it in terms of the actual etiology for it? Again, with genetic being one that we're going to talk more about, but that there are multiple different types of causes for epilepsy that we have to take into account here. The International League Against Epilepsy most recently came up with this as their conceptual model for for a way of classifying epilepsy syndromes. And I think that there's a number of advantages to this, even though this is a pretty crazy slide with a bunch going on here. I want to break it down a little bit so that people can understand a bit of what's going on. So for starters, you have seizure types. And this is different than epilepsy type in an important way. So seizure types is just what are the actual seizures that individual has. Some and many people with epilepsy only have one type of seizure, and it may be generalized tonic-clonic seizure, it may be a focal seizure. We'll talk a little bit more about what we mean by that. It may be unclear. We know that they have seizures, but we don't actually know yet how to describe them or where they're coming from in the brain. When you think about epilepsy types, this expands a little bit because you might have multiple seizure types. So you could have focal and generalized, or you still may not know exactly where they're coming from in the brain. The basic distinction here being focal seizures are things that start in one portion of the brain and then spread, whereas generalized seizures are things that spread very rapidly to the whole brain. And there's different types of seizures that you've probably heard terms for that I'll talk a little bit about in the coming slides. Alongside your seizure types and your epilepsy type, you have your causes or etiologies for your epilepsy, and you have your comorbidities. And again, comorbidities just mean what else is going on with them, and particularly what else neurologic is causing problems for that individual, if anything. And within the etiology realm, you have structural. What we mean by that is some sort of change to the actual structure of the brain. That could be an injury to the brain. That could be a way in which the brain formed a little bit abnormally from start. We have genetic causes, we have infectious causes, metabolic, immune causes, and then we actually have a lot of things that we're not entirely sure what the cause is. And I will say that some individuals fit into more than one category. So you can actually have more than one cause for epilepsy, not just a single cause. So when we're talking about focal versus generalized, we think about this a little bit from the perspective of the squiggly lines that we read in an EEG. And what I'm pointing out here is that the left is all our odd numbers and the right is all of our even numbers. And all of these lines basically just represent a portion of the brain, which you can see sort of mapped out in the bottom right corner here. Which part of the brain are we referring to with the labels that we have for each of these squiggly lines? And if you have an individual who has a seizure that looks like the left panel here, you can see that these spikes that start pretty early on that page go in all of the different brain regions at the same time. So we see the whole brain being involved very rapidly for this seizure type. That would be an example of a generalized seizure. On the other hand, on the right panel, you can see that there are some of these spikier waveforms that start really in a couple of areas and then spread slowly to involve other areas. And so what that means is it's starting in one part of the brain and spreading to other parts of the brain. When we say focal, I think it's important to realize that the older terminology that was often used was partial. So partial seizures or complex partial seizures really mean the same thing as focal seizures. So what types of seizures fall into one of these two different categories? In the generalized category, there's a number of different seizure types that are often discussed. Generalized tonic-clonic is one of the most common types of seizures, which is what everybody thinks of as a grand mal. Generalized tonic, the difference is that tonic is the stiff phase, clonic is the jerking phase. So generalized tonic seizure is really primarily the tonic stiff phase without as much of the jerking part. Myoclonic, on the other hand, is just the jerk. So a myoclonic seizure could be an isolated single jerk, or it can sometimes happen in a cluster of several brief jerks. Absence seizures are brief, short, okay, so brief like 10 seconds, maybe 20 seconds typically. Staring spells where an individual is speaking and it's almost like there was a Zoom glitch and they just stop and then they pick up and go right back to where they were talking. Atonic seizures are loss of muscle tone seizure. So these are usually called drops, or can be like cutting the strings on a puppet is another way that they're described. Individuals simply just fall. Focal seizures are really complicated. because the brain is complicated and focal seizures can look like almost anything depending on where they're coming from in the brain. This usually involves some sort of asymmetry from one side of the body to the other. So the right side or the left side aren't doing the exact same thing. That's one sign that it came from one part of the brain or another part of the one half of the brain or the other half of the brain. And a focal seizure can spread to involve the whole brain and generalize. Usually that results in a generalized tonic-clonic seizure. There's another type of seizure called an epileptic spasm, which is common in infancy and actually can have either a focal feature or a generalized feature, and so fits into this bit of an other category. Just to make sure we're all on the same page about what we're talking about with comorbidities, we're really talking about everything related to an epilepsy that's not the seizures themselves. Is there intellectual disability? Are there behavioral challenges, motor or language concerns, abnormal sleep? There could be a whole host of other additional things that I haven't named here that may be comorbidities that are other challenges that individual has, particularly neurologic. And when we put all of that together, the comorbidities, along with the seizures, we start to think about how we can kind of map that out. And this is sort of a theoretical model that's mostly relevant to pediatric onset epilepsies, where we could think of the normal sort of typical developmental trajectory as being the black line. And we could see a number of different categories of epilepsy syndromes that are deviations from that typical developmental path that are related to the seizures that that individual is dealing with. And so in the red line, you see a couple of different trajectories where the individual starts off on a pretty typical trajectory in terms of their development until they start to have seizures, and then they start to deviate, maybe just not quite keeping up. In some cases, it could be a plateau, or it could even be a loss of skills, something we call a regression. And those are all going to make us think about different types of problems, different causes, and different epilepsy syndromes. On the other hand, you could have individuals who, even before they started having seizures, had difficulty with their development and an abnormal developmental trajectory. And that is going to make us think about a little bit different set of conditions and syndromes compared to the red line or the black line. So again, we've come back to the ILE Epilepsy Classification. And while it's a bit daunting here, we can take the information we have for a particular individual, what types of seizures are they having, what overall type of epilepsy do they seem to fit into, more of a focal, more of a generalized, or combined. Do we think of a specific epilepsy syndrome that they fit into at that time? Or maybe we don't have that information yet. And at the same time, throughout the whole process, we're going to be thinking about what are comorbidities we need to address and what are the causes. This gets mapped onto a pretty crazy list of traditional epilepsy syndromes. And this is the kind of information that you can find on epilepsy syndrome or epilepsydiagnosis.org. This breakdown that I presented here is really dealing with age-based categories for these syndromes, neonatal or infantile, childhood onset, adolescent or adult onset, and some that can actually present at any age and can be quite variable. And I don't know that it's necessarily helpful for us to, one by one go through each of these different types of epilepsy syndromes. But my point here is more to realize that there are a lot of different epilepsy syndromes, and these are the things that we're trying to understand which patients fit best into which of these categories. And I think with that, I wanted to think a little bit or discuss a little bit about how a syndrome relates to a genetic diagnosis. And the two are not equivalent in most cases. And so I think we need to be a little bit careful. So SCN1A is the name of a gene. It happens to be the most common genetic cause of epilepsy across epilepsy as a whole, and is most well associated with a syndrome called Dravet syndrome. However, SCN1A can also cause a number of other epilepsy syndromes other than Dravet syndrome. And there's some other genes that can probably cause Dravet syndrome. So this is not a one-to-one relationship. And that makes this a little bit complicated when we think about what's the relationship between what we call the genotype, the gene change we find in an individual, and the phenotype, how that individual is presenting. And those two are not as closely linked as we would like to think in many cases. And so that makes it a bit complicated. And so we now are at a place where, as we are having more genetic diagnoses, we have to use two different bodies of evidence or knowledge to guide treatment. One of those knowledges comes from what epilepsy syndrome does an individual have, and what do we know about that epilepsy syndrome based on all the articles that have been published and observations that people have made over the last several decades. The other one, which is a less robust literature and is really something we've been developing over the last 20 years and much more over the last 10 years, is what do we know about individuals who have changes in this particular gene, and how does that guide what we're going to do in terms of treatment for that individual and for management of other comorbidities. So just to kind of give some sense of how big of a phenomenon this is a often quoted figure from a review paper that shows each of the genes that were described and when they were described over the last 20 years or so. This is from 95 to 2015. Each one of these sort of alphabet soup letters represent a particular gene. And you can see that there's 20, 30 genes on just this graph, and the trajectory of discovery of these genes over the last 20 years, which has really accelerated quite dramatically. And I will tell you that there's now probably more like in the hundreds approaching thousands of genes that are associated in some way with a risk of epilepsy. And so this has really exploded quite quickly. We also look at this from a little bit different perspective, where we can think about if an individual has particular types of seizures, and we discussed what some of these seizure types were earlier, generalized seizures versus partial or focal seizures, myoclonic seizures, or absence seizures, what is the risk that they actually have a child who has epilepsy? And we know the overall population risk for epilepsy is about 1%. And so having a parent who has some sort of epilepsy does actually increase the risk for their children, not dramatically, might move it to 2, 3, 4%, maybe even up to as much as 9 or 10% in some of these cases, but there is some increased risk. So this is one of our lines of evidence that even if we can't point to a particular gene, that an individual's overall genetics does influence the risk of epilepsy over time and over an individual's life. Another way that we can kind of think about this, and this is a bit of a complicated slide here, but the orange might represent particular genes like SCN1A that can have a very big impact. In other words, a single change in just one gene may be enough to result in epilepsy. On the other hand, in the sort of yellow color are all of the other 22,000 genes that an individual has, and the role that some changes in those genes may play in the risk of epilepsy. Each one of those changes by themselves may not be enough to result in disease, but they may make the chances of epilepsy more likely or less likely in a particular individual. And then this all gets overlaid with environmental risk factors. And that could be any number of things. We know that there's impacts of socioeconomic status on epilepsy. This could be nutritional. This could be traditional sort of exposures to certain things in the environment. So there could be a whole host of things that might fall under that environmental risk factor category of things. And so as these all get added up, if there is a single gene that has an important change in it, multiple genes, that's what we mean by polygenic, multiple genes that have small changes that by themselves don't cause a problem, but together increase the risk of epilepsy, and environmental factors all can combine together to influence an individual's risk of epilepsy. So within that sort of framework, we're talking about the etiology of genetic etiologies within this overall ILE framework for epilepsy. And I'm gonna go from there into a little bit of like, how do we think about that then and approach a particular patient? So when we're thinking about an individual in front of us, generally speaking, we're going to work through that sort of classification system, and we're going to start to fill in the blanks. What type of seizures does that individual have? What, if any, comorbidities do they have? Together, what type of epilepsy do they have, and what sort of etiology? And as we answer those things, we can start to identify an epilepsy syndrome and guide care, as well as potentially the cause of epilepsy and how that might guide our care. And that also leads to a bit of a complicated decision around whether or not we should actually be sending genetic testing on individuals. And I think it's worth just touching a little bit about this so that people have a sense of whether or not it makes sense to send genetic testing maybe in themselves or a loved one with epilepsy. I will also be honest that this is a rapidly evolving area of medicine where genetic testing was very rare a decade ago and is now quite common, particularly in pediatric epilepsy, but I think this is also gaining traction in adult epilepsy care. So which patients does it make sense to send genetic testing? Why would one want to send genetic testing? When should you do it? And how does it change management or potentially change management? I'm going to go through a couple of slides here to answer some of these questions. So there's a couple of statistics that help guide should we send genetic testing. And one of those is that we see with certain comorbidities and at certain ages, there's a much higher chance of us finding one of those single gene causes of epilepsy. Honestly, we're not really good right now at identifying those multiple genes or polygene causes of epilepsy. They're a lot more difficult to piece together in an overall puzzle because you do have 22,000 genes. What we are pretty good at is identifying those high impact single genes that by themselves can result in epilepsy if you have a change in them. And so under the age of 2, we see a much higher risk. At neonates onset of epilepsy, we see an even higher yield for getting genetic testing back that tells us an answer. We also see that with either intellectual disability or developmental delays, that there's a higher chance of finding a genetic cause. And something that we call epileptic encephalopathies, which is actually another way of sort of linking developmental delay or intellectual disability together to say that, you know, those with epilepsy are going to increase your yield. And those are just sort of 2 different papers or a couple of different papers that have looked at that. One of the ways that we can sort of plot this out, and this is actually from some of the data that we have at Children's Hospital Colorado, is based on the age of onset of seizures, you can see a different percent yield of sending genetic testing, with neonatal onset being the most likely, and then it decreases some as individuals have onset of epilepsy at older ages. Not necessarily dropping to 0, but dropping much lower. The difference between the blue and the orange is whether or not there is developmental delay in that individual or that individual is developing typically. So if they do not have developmental delay, in general, they have a lower chance of finding a genetic cause, but again, it's not 0. And so these are some of the things that we weigh when we're deciding, does it make sense to send genetic testing or not in a particular individual. Sorry, just a couple of things there. And then we can also think about this in terms of imaging. When we look at the structure of the brain, does that tell us anything? And what obviously jumps out here pretty quickly is that this center column, the orange column, has a much higher chance of finding a genetic cause than some of the other columns. So what's the difference between these? The blue column are individuals who have a seemingly normal MRI. We don't see anything that is structurally different on that MRI. Or we see an MRI with a clearly acquired change in the brain. And what I mean by acquired is this is something that happened to an individual after birth in most cases. They had trauma to the brain, they had an infection, lack of oxygen, something like that, that there was an injury to the brain that results in the abnormal structure at that point. That's different than this, what we call developmentally abnormal MRI, which is really individuals whose brains formed abnormally during development in the womb. And so that's a bit of a different category. And we see that the chances of finding a genetic result is actually much higher in individuals who have had some portion or significant amounts of their brain who have developed abnormally from the start, as opposed to being sort of acquired versus normal. And other is a similar sort of thing to normal or acquired that we don't need to necessarily get into at the moment. The other thing I'm going to make the point of is that we also have a list of things that are treatable causes of epilepsy. What I mean by treatable is not just that we can give an anti-seizure medication and try to control seizures. I mean that we can actually target the underlying problem. What is the underlying cause? And can we do something about that? The point isn't to memorize this list here, but just to know that we also have a list like this in our head of things that we want to make sure we find if it's potentially treatable. Another way that we're starting to think about this is we looked at 90 individuals at our institution who had a clear genetic cause for their seizures. And we asked the question, how many of those was there a precision medicine implication? What do I mean by precision medicine? I mean that knowing that they had that genetic cause changed the way that we treated that individual. And there's sort of a couple of bins on how that might have presented. One, actual treatments or medications that we're giving them. And what we see on the right is a list of a couple of different genes that are relatively common causes of childhood onset epilepsy, that we know that we're going to treat them differently if we know that they have that gene. So certain medications, certain seizure meds are more likely to work or less likely to work in certain individuals, or that there is another type of specific treatment available, like from the list on the last slide. The other ways in which we think that knowing a genetic cause is important is it may influence our decision about whether or not to pursue an epilepsy surgery for a particular individual. When they have some features that make us think that epilepsy surgery might work, but if they have a genetic cause and that genetic etiology is in all of the cells in their brain, that may not be as easy of a plan as we would otherwise think. And so that can give us insight about whether or not an epilepsy surgery is going to be successful for a particular individual. That's not to say that we send genetic testing necessarily on everybody before pursuing epilepsy surgery, but in certain cases where there's some questions around it, we might want to do that. The other implication is around other aspects of medical management. So this is not that it influences what treatment we use for their epilepsy, but it might be that that individual has other potential medical problems, even non-neurologic medical problems, that we need to watch out for because we now know what their diagnosis is. And so that influences their overall medical management, even if it's not influencing their epilepsy care directly. And so that is also one of our areas of sort of precision implications for knowing the etiology. And with that, I was going to move into a little bit about what do we think about where this is going, and why does this matter over the next 5, 10, 20 years in a way that we only see little bits of so far. And I think there's a couple of different things to think about here. When we think about genetics, some of you might have learned back in high school a long time ago, or may have blocked out of your mind because it was painful. This whole idea of DNA, RNA, protein. DNA is our genome. That is our instruction manual for building the machines of our cells that help our cells function, particularly neurons, as being the thing that we would care about in terms of epilepsy. Proteins are those little machines. Proteins just do the work of the cell to make the cells function properly. If you don't have a protein or the protein doesn't work correctly, then just like a factory has a machine that gets off, the factory can't produce the product anymore, the cell doesn't work properly if all the proteins don't work properly. The translator between DNA and protein is something called RNA. RNA has gotten a lot of attention recently because of COVID vaccines and the mRNA-based COVID vaccines. And really, RNA in this case is a way of getting from DNA, you process it a bit to RNA, that RNA then gets translated into protein and makes those cells. And the reason this matters is because there's novel therapies that are being developed for a whole host of different types of genetic epilepsies that can target some of these different areas. So in some cases, people are working on whole gene replacement. We are going to inject brand new genes into individuals to replace genes that aren't working properly. And that's called gene therapy or gene replacement. And there's a lot of complicated aspects to being able to do that and do that successfully. But there are a large number of companies that are developing that for certain epilepsies. This has been successfully done and FDA approved for other neurologic conditions, with the most notable being spinal muscular atrophy, which is a rare muscle disease of childhood that is fatal if not treated. And there are successful gene therapy treatments now available clinically. RNA has other opportunities for intervention, and there are treatments called antisense oligonucleotides. It's a complicated name that we usually abbreviate to ASOs, which are basically small bits of synthetic RNA instead of what our body produces as RNA, that we can actually write the code that we want to target to a particular part of our RNA so that we can change the way that we're processing to correct some sort of problem with how we are processing our DNA into protein. And those are also promising, some of which are used for treating other neurologic disorders already clinically, and several of which are in development for various types of, again, epilepsy syndromes. Angelman is one of the more notable ones here, as well as SCN1A, both of which have ongoing clinical trials in this area. The last one is that you can actually give the protein. So you kind of skip the instruction part right to the end and you give them the machine themselves. This is also called enzyme replacement. Enzyme's another name for type of protein. And one of the best examples of this within the epilepsy realm is for a type of disorder called Batten disease and a specific type called CLN2. And that is a problem where the cell's system for kind of getting rid of waste and getting rid of trash in the neuron is not working properly. You can think of it as like the dump trucks aren't showing up, and so you get an accumulation of trash, and that eventually kills those neurons, and it is a fatal neurologic degenerative condition. That there now is a way that we can replace the protein that's missing. It requires frequent infusions of that protein into the space around the brain in order to be able to do that. But it is a treatment option that was not available prior to 2017 when it was FDA approved. And this is a very rare type of epilepsy, but in this case, it can be a real game changer that this treatment is available. And so I just wanted to give a little bit of a overview that there's a lot of really exciting things that are coming down the pipe, and that we can see opportunities where we're going to be able to actually come up with really, hopefully, amazing treatments for epilepsy in the genetic epilepsy space over the coming years and decades. And with that, I figured I'd just kind of recap a little bit about what I've talked about, and then we can answer some questions if there are any. So when we're thinking about epilepsy syndromes, we're really just talking about a cluster of certain traits and how they relate to each other to try to say fits best with this epilepsy syndrome. That is not a perfect system, but it is a way of us having some guidance about what's most likely to help, what kinds of things do we need to watch out for, and potentially a bit about prognosis. So it's better than nothing. Genetics are becoming an increasingly important part of clinical care, particularly within pediatrics, but this is also pretty rapidly expanding into adult epilepsy care as well. That can have important management and treatment implications that will guide what we can do to find the best treatment for a particular individual. And in the future, we're hoping to take some very rare examples of things where we have precise treatments that really get after the fundamental problem and expand that to become a much more common treatment option across a variety of rare epilepsies rather than the very few that we currently have available. And with that, I appreciate everyone's time and the invitation to speak, and I am happy to take questions. These are my boys at the zoo here.